Hello, everyone, and thank you for joining today. This is the Dana-Farber's Department of Data Science's Frontier in Biostatistics webinar. My name is Erica Fike, and I'm the program manager for the department. If you have a question for the speaker, feel free to use the Q&A tab throughout the entire talk. Our wonderful faculty organizers will be moderating the questions for Dr. Trippa during the talk, so feel free to enter your questions at any time. I will turn it over to Svetlana for the introduction.
All right, thank you very much, Erica. Thank you everyone for joining. Today is my greatest pleasure to introduce Dr. Lorenzo Trippa, our speaker today. He's a member of our department. He's an associate professor here at the Dana-Farber Department of Data Science, as well as the Department of Biostatistics at Harvard School of Public Health. Dr. Trippa graduated with his PhD in statistics from University of Bocconi in Milan in Italy. His research for his thesis work was done at he did it as a visiting scientist at the MD Anderson Cancer Center, and then he joined Dana-Farber. He's associate professor, as I mentioned, and he's also a co-director of the program in regulatory science, where they work hard to bring the most innovative clinical trial designs to our patients. And Dr. Trippa is interested in Bayesian statistics, novel clinical trial designs, and he will tell us about it to us today. Thank you.
Thank you so much, Svetlana, for the introduction, and thank you to all the organizers. It's great to have this opportunity to present our work, and the goal is to understand how can we leverage external data sets that we have, and the goals are both the design of future clinical trials and also deciding what are good approaches for data analysis. So the first slide is just a graphical representation of three elementary, very simple trial designs. And on the left, I have a single arm design. So every patient enrolled in the study is assigned to the experimental arm. The typical goal is to understand if the treatment has a positive effect compared to the standard of care. And the information on the standard of care relies, in most cases, on publications, point estimates of the response probability or of the survival distribution. Then, if we look on the right, we have the standard randomized clinical trial design. Here I have one-to-one randomization. I keep it as simple as possible without interim analysis. They provide very different level of evidence. The techniques, approaches that we have to design them and to analyze are quite different. Here, randomization trying to address the problem of possible confounders. And in the middle, there's an intermediate solution. Again, I have a single arm, but I use external data set with patient level data. And that means that I have information, not only historical information on the outcomes, but also on the individual pre-treatment profile, so clinical variables that are relevant for the analysis. Main difference between single arm and single arm with an external control is the type of analysis that I can do. In the second case, I can apply methodologies from causal inference literature that have been developed for observational data. So what is the key question in this slide, and what I'm going to try to discuss in the talk? As biostatistician, we don't only need to provide the details and the sample size of the design, but we need to choose among different options. Here I have three of them, and they're very different, both in terms of assumptions. Single arm design assumes that there's little variability when I look at populations in different trial studies. The methods in the analysis is very different. Resources the number of patients that I need in order to implement different designs can be substantially different. And obviously, the risks are markedly different. Here I'm pointing specifically to risk of bias in the treatment effect estimation, as well as poor control of operating characteristic, and in particular, possible type 1 error, so high rate of false positive results. So how do we choose one among different trial designs? And the second slide, it emphasizes a different aspect. And here I want to emphasize that there are very different ways that I can use external information. So if I look at the top of this slide, I have a graphical representation of a randomized trial designs. And those yellow bars are just interim analysis in which the interim decision is to stop or continue the trial, and we can stop for futility. So that means during the trial, we can decide to either leverage only the internal data that come from the randomized trial and make these decisions. Our option B is to combine that early data with external information that we have from the clinical trial. And here I'm keeping it conservative. And when you look at the right bar in the graphical representation, that's the final analysis. And I'm assuming that the final analysis use only the internal data, randomized clinical trial. So that's a design that can be useful for regulatory purposes. Indeed, we don't have any possibility to inflate the type 1 error beyond what happened in a standard RCT design. When I look at the bottom, there's another design. In this case, for the phase 2 setting, I have an initial stage of the trial, which is one-to-one randomization to the experimental and the control arm. Then in the middle of the trial, there is an interim analysis. And the key purpose is to understand if the external data are appropriate. We want to understand if there are substantial discrepancies between the external data set and what we see in the control arm, in the early data of the control arm. At that point, we can make a decision to continue with one-to-one randomization. Or if we are convinced that the external data are reliable, at that point, we can decrease randomization. In the extreme case, we can discontinue randomization and enroll in the second part of the phase 2 trial patients only to the experimental arm. This is what we call a hybrid solution. And here the advantage is that we can try to reduce the error in the estimation of the treatment effect. So what do we try to do is the following. Well, we start looking at what happened in glioblastoma. And there's a key advantage. The standard of care has been the same or with minimal changes during 15 years that allow us to build our literature on a number of phase 2 and phase 3 clinical studies. And in addition, we can have patient level data from some of those studies. And we want to use them in order to evaluate these designs, these options to leverage external data. A key question is, are we going to bring in the trial too much bias? Is there a relevant risk of losing the control of type 1 errors? What is the trade-off with the resources that we need in order to implement these designs? So the idea is to build on previous data. And the next 3-4 slides, I just want to give a summary of what was the key motivation for doing this work. 3 years ago, we started Insight. I designed it using a Bayesian adaptive randomization. I'm not going to discuss any detail on the modeling and on the adaptation of the probabilities, But just mentioned, the idea is to have the biomarker profiles before randomizing each patient. And then we'll look at two outcomes, both progression-free survival and overall survival. The goal is, first of all, try to match each patient with those arms that are more likely to give them a benefit. And the second one is to try to accelerate enrollment on those arms that are more promising. And I want to mention two characteristics of the trial. Here's the first one, very simple, but I think it's the most important. During time, we allow to add new arms. And actually, we are in the process of adding two more arms, more experimental arms to InSight. And what is the benefit? The benefit is that we don't want to replicate over and over, as you see in panel A in this slide, the same control. Here, the control is in blue. And instead, we want to have a platform, multi-arm design, where the control arm can be extended and we can add and remove experimental arms. So we don't want replicates in the resources, just to have a good representation of how patients respond under the control. Second characteristic, as I mentioned, why do we use progression-free survival and overall survival? Well, we use them with two different goals. The overall survival is our primary endpoint, but PFS has the advantage of giving us information much earlier. And we need early estimates both of treatment effect. We compute the posterior probabilities that each arm has a positive treatment effect, and a posterior probability that the trial will be able to demonstrate that that benefit. So that's the core of the adaptive randomization. And that's why we have a joint modeling of the two outcomes, which are used for assigning patients and for final analysis as a primary endpoint. And I want to mention this decision is based on the fact that in most studies we expect correlation between the two outcomes, but there have been important exceptions of treatment that showed marked positive effect on PFS, which then didn't translate at all into overall survival benefit. Here's just one slide taken from a recent presentation of early results of abemaciclib arm of InSight. I will not discuss this part, but we are at the stage where we are already completing enrollment on some of the arms, and we will have the results soon. Now, I want to go back to the primary topic of my presentation, which is how do we translate previous experience data from completed clinical trials into good decisions, not only on sample size and other relevant aspect of the design, but how do we choose whether to incorporate external information or not? How do we choose to randomize or not? This is what I'm gonna focus. This was the first step that we took in that direction. This is a meta-analysis, and we looked at all trials during the last 15 years. We have around 417 studies, phase 2 and phase 3 trials in newly diagnosed glioblastoma. So, and here, the key information is what is the typical design that is used in this disease? What are typical sample sizes? How many resources are allocated to the phase 3 confirmatory stage of experimental treatment versus early studies? And we have this is part of the results that that we got from that meta-analysis. This table has a very simple interpretation. It is a list of all phase 3 trials in a period of time of 11 years. And all of them failed except the TTF, which is not a drug, it's a device, and it's still largely controversial. It's not adopted by the majority of the clinicians. So we see a clear pattern. We see such a number of patients allocated to confirmatory trials that then don't translate into an improvement of the standard of care. And this suggests an hypothesis. An hypothesis is maybe the phase 2 designs are not appropriate. Maybe there's something wrong in the interpretation of the data from phase 2 studies, which then are used to motivate the design and the start of a large phase 3 studies. And what we see is that when we look at the phase 2 results and designs, we see largely single-arm studies, and very frequently we say that they look at PFS while the subsequent phase 3 is looking at improvement of our overall survival. So we ask ourselves, is there a problem on these decisions that come from single-arm studies? And we use a very simple modeling approach. So the title of this manuscript is to randomize or not to randomize. So that's the key question we try to address, and we try to address specifically for GBM. This is a random effect model. So what I indicate as P0, it's the response probability when we look at the average across different trials for the control treatment. And the same control treatment can have different response rates across studies, just because they enroll different patients or because there are different in the management of the disease. That's why we have this random effect in the error. And here the subindex T indicate one specific trial. So the larger the variability of that random effect, the more we should be concerned about running a single-arm study. And indeed, the single-arm study is based on the assumption that responses under the control are highly predictive. The distribution of the responses is highly predictive for the control. And if sigma 0 is large, that's not the case. Then we have two other key parameters here in red. One is delta. So that's the measure of the treatment effect that we hope to be able to demonstrate. And the other one is beta. So the single arm analysis involve an estimate of how patients would respond under the standard of care. And that estimate can be wrong, it can be biased. In GBM in specific, we think it's been biased in several occasions because an old trial was used as a reference point, as the point estimate of P0 was taken from the first study that looked at the combination of radiation therapy and temozolomide, which is the current standard of care. So these are key parameters, and depending on the value of those parameters, it can be appropriate to utilize resources for early study, allocating them to single-arm trial. It might be completely wrong. That's the case if sigma 0 is large. Okay, so the nice thing is that we can estimate P0, beta, and sigma 0. And in fact, we have randomized clinical trials, and we can look at response rates for their control treatment. And here you see all of them use the same control, temozolomide plus radiation therapy. So we have replicates of different studies that look at the same control and report on the same outcome. Then we also have single-arm trials, and we can look what do they use as a benchmark? What do they use for a point estimate of the efficacy of the standard of care? And most of them have the same approach. They take the point estimate from the first paper published on temozolomide plus radiation therapy, confirmatory study. And we think that that is an underestimation of the efficacy on the current population, just because the management of the patients changed during 15 years. So now, what can we do? We estimate P0, beta, and sigma 0. And here I want to emphasize the approach to estimate those parameters is very, very simple. It's based on just fitting a random effect model. And at that point, we can look at the operating characteristic of a single-arm trial and a randomized clinical trial. So here, we assume that we have a fixed amount of resources. We can enroll only 60 patients. And what this figure tells us as the following. If my primary criteria is just mean square error, then since our estimate of the standard deviation, what it's indicated as sigma 0, is above 0.6, it happens that the single-arm trial would have a larger mean square error in estimating the treatment effect compared to the randomized clinical trial. So this is the logic. We use previous clinical trials in order to estimate the operating characteristic of various options. Here we have two options, single-arm trials and randomized clinical trial, and we look at a specific metric, mean square error. But I think this is a metric that is more relevant, especially for the early stage clinical trials. We do exactly the same type of exercise, but this time we don't look at mean square error. Instead, we look at the type 1 error probability. And what this panel tells us is the following. Sigma 0 estimated from our clinical trials is so large that even if I'm able to have beta equal to 0, even if there's no bias in estimating the average response rate across populations, the standard single-arm trial with off-the-shelf analysis has a type 1 error that it's around 20%. And that's aligned with what we see later in the phase 3. So many failures in the phase 3 setting after single-arm studies in the phase 2 setting. Second part of the presentation, I'm going to introduce the use of external data set. And I start from this graphical representation, which indicate our effort in trying to validate, trying to understand if it's a good idea to use external information or not. If you look at the right part of the slide, this is the probably the simplest approach that we can utilize. We take patient level data from a randomized clinical trial, here indicated as RCT1, which compared a control arm and an experimental arm. And I'm going to use it as a benchmark. So the treatment effect, I'm going to assume that is unbiased. and that I can evaluate the variability of that estimator. But then what I do, I utilize only the experimental arm data combined with external data sets. So I don't have any more randomization. Here, I am producing an in silico trial with smaller sample size because patients are only on the single arm trial. And then I use adjustment methods, which are taken from the causal inference literature. So in this very simple validation exercise, what I hope is to see a strong alignment between treatment effect estimate and inference in general on the treatment effect. In the RCT trial, which I consider the gold standard, versus the other design, which combine single arm and external data. If that is the case, and if that result can be replicated for several randomized clinical trials, I can start saying that I have some signal telling me that the idea of a single arm trial that is controlled by an external data set, it's plausible. If instead I see strong alignment, then that indicates that there are problems, and problems can be, for example, missing confounders, so relevant pre-treatment variables that are not measured or that are not measured accurately, and other issues that that we can have in this schema is that outcomes might be measured in a different ways in the trial versus the external data sets. So here, another validation schema, and that's what we used in this manuscript by Stefan Benz. We found it particularly useful. You see, it's very similar to the previous slide, but in this case, we don't isolate the experimental arm of RCT1. We isolate the control arm of that randomized clinical trial. And what is the advantage? The advantage is that those patients are under control, and they will be compared with other patients from different studies, from different data sets, here indicated in green, that received exactly the same treatment. So in this validation schema, we hope that the adjustment methods will tell us, oh, the treatment effect is close to zero. And if we verify that repeatedly for several trials, repeating this exercise of validation, then this supports the idea of an externally controlled trial. So the goal obviously is to understand if we have enough information on pre-treatment variables for the patients, and if there is or there's not elements that can distort this comparison of patients enrolled on the experimental arm versus the external control. So summary of this manuscript, we compare 3 different trials, an RCT, single arm trial, and this is a single arm trial that used the ERTC estimate. ERTC is the first trial that developed temozolomide plus radiation, the current standard of care. And the third option is the externally controlled single arm trial. So where we integrate external data using propensity score, inverse probability weighting. We have various options that we considered. The metrics that we looked at is bias, variability of treatment effect, possible deviations for the type 1 error, the possibility of controlling accurately false positives, and the sample size that it's necessary to achieve a targeted power. So very interpretable metrics of efficiency. And so this table is the data collection that we used in the analysis. 5 studies, all of them involved the use of the same standard of care treatment, temozolomide plus radiation. 2 are cohorts from DFCI and UCLA, and the other 4 are randomized clinical trials. You see we have a list of variables that were available in these studies, including age, Karnofsky, performance status, and a few relevant biomarkers in GBM. So the key question is this sufficient in order to do our adjustments, or do we have a measure confounder that compromise this single arm with external control design? And this is just a table that confirmed that at very minimum the variables that are available correlates with primary outcome. And here, well, this slide is the main tool that we used in order to understand if the operating characteristic of ECT externally controlled trial are reasonable or not for phase 2 in GBM. What we do is the following. We isolate one clinical trial and we subsample only the control arm, temozolomide plus radiation arm, and we have information for those subsamples X and Y, both pre-treatment profiles and outcome. Then the remaining 5 studies constitute our green block, the external data. And then at that point, we can use off-the-shelf methods from causal inference in order to estimate treatment effects, in order to test if there is a treatment effect or not. But these are trials in which we actually know the treatment effect because we are sampling from the control arm. So we know the treatment effect in this exercise is equal to 0. And we know the H null should not be rejected. So by doing this iteration, we evaluate if the design has appealing operating characteristics or not. This is the main figure of the manuscript. What happens when we use n equal to the actual sample size of those 5 data sets that I summarized earlier? Well, we know treatment effect is equal to 0. If we implement the single arm trial design, which compare with a fixed benchmark, a fixed estimate of what I called P0, then we have 2 false positive results. And the result change when we look at those blue bars that are representative of inference that we would have with externally controlled trials. See, in this case, there is a shift, less bias in the treatment effect estimation, and those 2 false positives disappear. And here, very similar exercise within the same project. But what we did was exploration of many different approaches offered by causal inference literature. This is the y-axis of these graphs. And actually, we didn't observe much difference, both in the point estimates across methods and in their variability. So the key result when we subsample 46 patients is that we have a markedly different control of type 1 errors if we look at externally controlled trials compared to single arm studies. In the single arm study case, out of 10,000 subsamples for the various studies, we see that the false positives can be as high as 40% when alpha is equal to 0.1, and we don't have that problem. That problem is solved when we apply this list of different adjustment approaches. This slide tells us, well, now that we have learned that externally controlled trials in GBM are likely a better approach than the single-arm trial with comparison to a fixed benchmark, then we looked at the comparison of ECT and randomized clinical trend. Here, what I want to emphasize is that there is a substantial difference in the variability of the point estimate of the treatment effect. So there is an advantage in terms of efficiency. This last slide on this project confirmed the same point, because we say that the ECT, which is illustrated in blue, not only it solved the problem of controlling the type 1 error, but it has a similar power of discovering drugs with a positive effect, very comparable to the single-arm study. And it's for standard sample sizes for the GBM setting, we have an advantage which can be in the order of 20% of the power. So we have an increment of power when we compare to the RCT design. The last part of the presentation, we try to expand and to consider more designs, more approaches to leverage external information. And here we have the comparison of studies indicated as A and B. They are both randomized clinical trials. Both of them include futility analysis. Option A is standard futility analysis that leverage only the data that are generated from the trial, internal data. Option B, when I do futility interim decision, I integrate internal and external data. So there's an augmentation of the data that are involved in the decision to continue or discontinue the trial. And I mentioned this decision are based on posterior probabilities. So we do Bayesian modeling of the data. Option B, joint model. See that the probability that will reject the null hypothesis below a certain threshold, then we indicate as appropriate to stop for futility trial. Something that I want to emphasize is that in this comparison, both option A and option B have identical final analysis, and this final analysis is just a Z statistic procedure to indicate whether there is a treatment effect or not, and a standard procedure also for the point estimate of the treatment effect. So the difference is only at the level of interim analysis for futility stopping. And the futility stopping component is completely ignored by the final analysis of the trial, as we see in most both phase 2 and phase 3 trials. So there is no adjustment for interim analysis. It's not necessary when we stop for futility. Of course, it would be highly necessary if we can discontinue the trial also for efficacy, but that's not the case here. So in this project, which was recently accepted by Neuro-Oncology, we have an evaluation that is based both on simulations and on a collection of data sets. So in simulation, we consider scenarios where there are missing confounders and measure confounders. We consider various distortion, as well as situations where our external data allow us to do accurate corrections, accurate adjustments. Well, the results are aligned with what can be everyone's expectation. If there is an alignment between internal and external data, In an ideal situation where we can do adjustment as expected, we see that we can reduce the average number of patients enrolled in those studies that are evaluating a drug without a positive treatment effect. So we can stop earlier those studies. And well, something interesting is also that if there is a treatment effect, we have exactly the opposite. It happens that incorporating external data can have an advantage and can reduce the probability of stopping the evaluation of a drug with a positive treatment effect. And the nice result is that we have an increment of the power just because we don't discontinue the evaluation of that drug. So here is just a table. I will not go through the details, but these are the primary results. If there is no unmeasured confounders, we can have an advantage both in terms of stopping earlier trials by integrating external information and reducing the probability of stopping the trial, which in turn translate into an increment of the power. And we also illustrate scenarios where there are unmeasured confounders. If that is the case, then we are in trouble because we can lose the control on those probability of stopping trials, and the result can be stopping a treatment with a positive effect, or it can be losing the ability of stopping treatment without treatment effect, or potentially even with inferior compared to the standard of care. Here, one slide just to emphasize that exactly the same subsampling algorithm that I discussed earlier can be adapted to this analysis. So we want to evaluate if it's a good idea to integrate external data in interim analysis. And what we do here, we isolate the RCT1, we subsample the control. That means we're gonna generate trials where the treatment effect is equal to 0. And then we use the other temozolomide plus radiation groups in order to form our external data set. And then we compare a plan of interim analysis with and without the external data set. And of course, also in this case, we repeat the same type of subsampling procedure thousands of times, not only considering subsampling from RCT1, but from all of the study. And then from that, we try to learn and to have estimates of the operating characteristic of the design. Here, a summary of the result for this project. We have consistent results when we subsample the various data sets, the FCR, UCLA, and the Evalio study, which is a large trial that enrolled more than 300 patients. These estimates tells us that in GBM, we are in a good position to integrate external data for early stopping. In all cases, we see that if there's no treatment effect, we have an increment of the probability of stopping soon the study. And then we looked what happened when there is a treatment effect. Well, the treatment effect, the positive treatment effect case, it required us to spike in treatment effects in the subsampled experimental arm. And that can be done quite straightforwardly. If we have time to event outcome, we just extend or multiply by a factor above 1 the type 2 events in order to produce a positive effect Here, in this specific case, we are looking at a binary outcome. And what we do, we relabel a fraction of negative outcomes into positive only for the experimental arm. So what happens in all three studies, if there is a positive treatment effect, we see that under the procedure that that we propose, which is interim analysis, leveraging external control data and integrating X, which are pre-treatment profiles, we have a reduction of the probability of early stopping, and that's a desirable characteristic. And in all three cases, we have an advantage in terms of power, so the power increase in all three cases, when we compare to a standard plan of interim analysis that is based only on internal data generated from the trial. Okay, so in the last couple of slides, I want to mention these results are specific for glioblastoma. And I want to quickly go through a more difficult exercise that we tried in lung cancer, where we explored different designs. The idea was to try to modify the randomization probabilities during the trial based on interim analysis that inform us whether the external data are appropriate or if we have missing confounders. I will not go through details of the various design. I will just say that we were in a situation where the pre-treatment profile of patients across the various data sets that we tried to utilize were much more incomplete compared to the application in glioblastoma, and the results are negative results. So this figure just tells us that for some of the studies that integrate external data, we had clear information. It's not a good idea to rely on those data sets just because we think there are problems of missing confounders. So this complete my presentation. I want to give a very brief summary. The main point I want to make is when we have multiple options in terms of designs, very simple decision like a single arm versus a two-arm randomized study, or a more complex option that involved the integration of external data. In both cases, we can try to utilize both data from the literature and patient-level data sets in order to guide this decision. And the decision can be guided by estimates of the operating characteristic of candidate designs. The second point I want to emphasize is that external data sets are not only useful for understanding in very early stage trial if there is a treatment effect or not. We can use them for several other purposes. I illustrated the case of interim analysis with futility decisions, but we can utilize us for other purposes, early stopping for efficacy, or for looking at other approaches, a combination of treatments, or a phase 1 trial that try to optimize the doses. So there's really a galaxy of options where external data can be useful. And the last point is the limitation of the approach. So how much do we rely on this validation? Well, a lot is driven by, here indicated with capital K. That's the number of studies for which we are able to have patient-level data. It really doesn't matter too much the overall number of patients that are included in our collection of the datasets. Our ability to understand if there is substantial study-to-study variability in the enrolled population from different studies, and to learn if there are missing confounders or other distortion mechanisms. That depends a lot on the number of studies for which we can have data. Very last slide. So the first authors, I have the two papers that I discussed, Alyssa Vanderbeek and Stefan Wentz. I want to thank them. And I want to thank also all the other collaborators, and I thank you for your attention. Thanks.
Thank you very much, Lorenzo. It was very interesting. So if anybody has any questions, please post them into Q&A section. And while we're waiting for the questions, I wanted to ask you, so as you say, so the relevant metric is the number of the studies that you can get the patient level data. So could you tell us a little bit about the state of the affairs in that? How easy is it to get? Is it common that now people provide this information? So as far as I understand, Project DataSphere is trying to get a little bit of that data out there. So can you comment on that?
Yeah, so I mean, short answer. It's very difficult to get more than 3 studies, because you need studies that use the same control. Well, that helps quite a bit, not strictly necessary, but definitely you want them to look at the same population, and you don't want probably to include studies that are 25-year-old, because otherwise you lose the comparability. And in many cases, we see that it's difficult to have access to those data, both for government-funded or companies-funded trials. There's huge literature on how it's important to share data, and my experience, it's still very difficult to do that. So one hope on this, I think that meta-analysis, useful and interpretable, can be based on a mix of patient-level data and data summary from the literature. If we are able to utilize both of them, then we can easily increase the K value and our ability to understand study-to-study variability.
I see. Thank you very much. And maybe when people get more and more familiar with all these methods and how it's helpful, maybe they will be more willing to share the data.
I hope so.
Well, we'll see. So we have a question from the audience. So could you please share the references to the papers that you're mentioning here, or tell where to look them up?
Definitely. So the first paper that I presented, this one, is published on Clinical Cancer Research. And here's the reference. It's Clinical Cancer Research 2019. First author is Stefan Wentz. The paper on randomizing versus not randomizing, it's on neuro-oncology. The title should be To Randomize or Not to Randomize. Then we got a paper just accepted, and it will be on neuro-oncology. The title will be The Use of External Control Data for Predictions and Interim Analysis in Clinical Trials. It's not yet online. It was just accepted. If you email me, happy to share it. These are the manuscript that I went through.
Thank you very much. So last call for questions.
This is Ashmay. I have a question. Thank you very much. Very nice talk. So my question is about the external data, right? So how you determine if the including external data is reasonable? I think if the external data is pretty much very much heterogeneous, I think it doesn't make sense to include that. How you determine if this external data is reasonable to include? Is there any criteria or something?
Yeah. So, I mean, two parts of to answer. First, there are a sequence of very reasonable steps. So looking at the literature, having an idea what are key predictors of the outcome and key variables that might influence the enrollment into trials. Trying to understand what are possible mechanism of selection bias in the external data. And try to verify that the data dictionary of different data sets is aligned. What we call PFS in one study can be quite different from PFS in another. The time clock can start at randomization or at a different time point when we look at different studies. So these are relatively simple steps. In my opinion, they are not sufficient. In the end, you need to declare what is the study, what is the design, including the analysis plan that you want to consider. And then apply that design to control arms. And if you see reasonable operating characteristic, if you see that your estimator doesn't look to be biased, if you see that the control of the type 1 error seems reasonable, then you can continue the development of that design. But otherwise, there's a clear signal that the external data are not appropriate.
Mm-hmm. Well, thank you very much. That's very helpful.
Well, thank you so much, Lorenzo. It was a great pleasure to listen to your lecture.
Thank you, everyone.
And let's hope that more and more data becomes available so everyone can design their trials using these methods.
Thank you, Svetlana. Thank you, everyone.
Thank you.
Thanks, Lorenzo. That was great. Bye.
Thank you. Bye.